Pues muy buenos días a todas y todos. Es un placer y un gusto enorme estar esta mañana aquí con ustedes en el Hospital Infantil de México, y para mí es un honor agradecer de parte de la doctora Rosana Pelayo, aquí presente, de la Unidad de Educación e Investigación del Instituto Mexicano del Seguro Social, y de mi parte de la Coordinación de Investigación en Salud del Instituto Mexicano del Seguro Social, la oportunidad que nos dan para que se dé el día de hoy ese seminario que para nosotros es muy importante. Quiero agradecer de manera muy especial a la doctora Mara Medeiros y al doctor Rafael Vázquez la oportunidad de que esta mañana nos den la oportunidad de que esté con nosotros el doctor Clive Drake. El doctor Ezequiel Fuentes Panamá lo va a presentar en un momento y nos va a hablar un poquito de lo que ha hecho y su importancia, pero yo nada más quiero enfatizar pues el agradecimiento de que esté esta mañana con nosotros. Él está afortunadamente en un sabático con nosotros en el Instituto Mexicano del Seguro Social, y ha sido de manera pues muy importante su presencia, y lo que queremos esta mañana pues es compartir y hacer una alianza importante estratégica aquí con el Hospital Infantil para poder establecer lazos, para seguir haciendo eventos conjuntos, para seguir fortaleciendo la investigación en conjunto y pues para poder hacer futuras colaboraciones con la presentación del doctor. Entonces, sin más, el doctor Fuentes Panamá va a presentarlo y al final vamos a tratar de dar algunas palabras más de agradecimiento. Muchas gracias.
Hola, buenos días. Es un honor recibir como invitado de este hospital al doctor Clive Maurice Gray, profesor de la Universidad de Stellenbosch y profesor emérito de la Universidad de Ciudad del Cabo, Sudáfrica. El doctor Gray tiene un doctorado en inmunología por la Universidad de Witwatersrand y realizó estudios postdoctorales en inmunopatogénesis del VIH en la Universidad de Stanford. Con más de 30 años de trayectoria en el tema, sus contribuciones han sido fundamentales para comprender las consecuencias de la exposición intrauterina al VIH y a la terapia antirretroviral en recién nacidos no infectados. Actualmente estudia los mecanismos que vinculan la terapia antirretroviral, la disfunción placentaria y los desenlaces adversos al nacimiento, que es justamente el núcleo de la conferencia que escucharemos hoy. El doctor Gray además preside la Federación de Sociedades de Inmunología Africanas, encabeza el área de educación de la Unión Internacional de Sociedades de Inmunología y fundó Inmunopaedia, una importante plataforma de formación en inmunología para el sur global. Actualmente se encuentra en una estancia como profesor invitado en el Centro Médico Nacional Siglo XXI y en el Laboratorio de Citómica del Cáncer Infantil del CIBIOR en Puebla, donde impulsará nuevas colaboraciones en salud materno-fetal y oncogénesis fetal. Si comparten intereses de investigación, les invitamos a acercarse al doctor Gray al finalizar la presentación para conversar e identificar posibles oportunidades de colaboración. Recibamos entonces con muchísimo entusiasmo y un fuerte aplauso al doctor Clive Gray. Bienvenido.
Thank you very much, everyone, and it's a very great pleasure for me to be here in Mexico and in Mexico City, and I've seen your beautiful country outside of Mexico City as well. So I'm also having a very intellectually rich experience with Dr. Rosana and others here at IMS next door. So let me get my... There it is. This is Cape Town. So some of you may have been to Cape Town, I know that for sure. And I'm going to talk to you about some of the work that we've been doing over the past 5 years and why we're doing it. So this is where I'm from. This is my institute, the Biomedical Research Institute. And I formed what we call RIACA, which is the Reproductive Immunology Research Consortium in Africa. And maybe we should change this to the consortium in the South, because part of me being here is to seek collaborations here in Mexico. So this is my group. We're outside my laboratories on the 5th floor of that nice looking building. The people on the right here are not in the picture, and I just want to shout out to Brandon, who's a very talented PhD student of mine, who has generated some of the data that I will show you. And also Michael, we have implanted him in our labor ward so he can collect the placentas, which is very important for a laboratory that is not a clinical laboratory to do that. So here again is Cape Town. We hosted, as you might know, the World Cup, as you did, but 16 years ago. And this is the legacy of the World Cup, this, if I may say, a white elephant here on the beautiful side of the mountain. But if you go the other side of the mountain, you find a very slum-dwelling, poverty-stricken environment where unemployment is almost 100%. People are extremely poor, and it's where tourists never go to, but you see it when you land in the aeroplane. And so we have our research sites in two areas. This is one of them, Khayelitsha, which we call townships. And the other one is Guguletu, and here is the airport as you come in. And our research building, that very nice looking building, is just about here. It's quite close to these sites. So what is the problem that we're facing in South Africa? The problem is, of course, HIV infections. And what I'm showing you here is the very bloated world mapper of Southern Africa, and the darker red, if you can see it here, is South Africa. And if you look here at the proportions of new infections, these are new infections, you can see that South Africa forms the highest piece of the pie. So the problem we're facing is when you look at the prevalence in adults aged 15 to 49, which is only a snippet of those who might be infected, we're looking at greater than 15% of the general adult population. If we home in on age distribution and we look at the breakdown by age, and what I've done here is I've boxed the childbearing age of women. And you can see straight away, if you look at the orange bars, that between the ages of 35 and 39, there is the highest prevalence of HIV. So this is not incidence, it's prevalence of HIV, which really translates to 1 in 3 pregnant women attending antenatal clinics being HIV infected or living with HIV. Thankfully, they are on antiretroviral treatment, which is a very, very fantastic program by the South African government, and it's free antiretroviral treatment to anyone who's living with HIV, including males and females. But of course, the childbearing ages may fall within this age and may fall outside. But this is the problem that we're being faced with. So with every success, because antiretroviral treatment has mitigated or reduced HIV transmission from mother to child to certainly below 1%, even closer to 0.1%. But with that comes a caveat. And the caveat is that these drugs are not benign. They probably have a complication around the vasculature development, probably most likely, which is what we're looking at in the placenta. But I won't talk about that today. What I'm going to talk about are 2 viral infections, and the other one I'll come to. So you can see here that, that South Africa has the highest prevalence of what we call HIV-exposed, uninfected infants or children. So they're exposed to HIV at some stage and antiretroviral drugs at some stage. So what is very important to, I think, for me to relate to you is that antiretroviral treatment is provided at no cost. It's either given to women prior to conception or during gestation. But either way, these children are HIV, what we call HIV-exposed, uninfected. So the issue here is that these children are not healthy. So they're not infected, there's no HIV, but they've been exposed to something in utero. And so there's plenty of evidence to show that there's greater odds of stunting. They have increased morbidity. Thankfully, with the lower transmission rates, mortality has fallen, but these children still die too soon. Lower birth weight. They have poor expressive language and motor development by the age of 2. There's quite a bit of literature on that. Lower working memory, and there's also residual inflammation. And what we believe is happening is that there's inflammation, maternal inflammation being transmitted. It may not be the virus, but it's the maternal microenvironment that is being transmitted in utero to the developing infant. So we published a few years ago, we've been trying to find an immune signature in these children for about 10 years of these HIV-exposed, uninfected children. So this is one signature that we found quite significant. So we looked at the types of memory cells, the types of T cells, the types of innate immunity. Nothing really convincing, but what popped out very convincingly was this, that there was a premature skewing of the T cell receptor and a delayed memory expansion in these HIV-exposed infants. And that was the one telling point of what we could identify. And in summary, what we found when we look at the T cell receptor clonotypes, interestingly, in what we defined as the naive CD8 T cells, there was, in red, you can see the HEU. There was a lower inverse Simpson index, which means that there was less diversity. Now, you need great diversity of your T cell receptor to interact with all the thousands of epitopes from the different pathogens that the children are bombarded with after birth. But interestingly, there's lower diversity in the naive population. In the memory population, this was expected because it's memory. But interestingly, there was a large proportion of memory T cells at birth. And this is birth blood. It's not the cord blood. It's within 6 hours of birth from a heel prick. You can't really see this very well, so you're going to have to believe me. So what we found is, let's orientate you. This is the memory compartment. This is the naive compartment. What we're doing here is we're predicting the numbers of T cell receptor interactions with the epitopes. So it's not specifically biologically what they're reacting with. It's a prediction. It's an algorithm, if you like, an AI platform. And what we find is in the HIV unexposed, uninfected, what we term as our controls, had lots and lots of these naive cells recognizing epitopes, predicted epitopes, and some in the memory compartment. But there was nothing in the HIV-exposed, uninfected. Now, you're going to find out in a moment that I'm going to talk about cytomegalovirus, co-infection with HIV. And many of these dots, and the size of the dots represents the size of the clone, are recognizing CMV. But the point here, and the conclusion we're coming to, is that maybe the absence of any clonotypic recognition of epitopes lays the path open to vulnerability to some sort of infection. We also published, and I'm just giving you this as background, some data showing that in the absence of transmission of virus, you still had a T cell homeostatic imbalance when we looked in the placenta. So the premise here is, well, let's not look in the birth blood, let's look before that. What's going on in utero? Because that skewing or the absence of clonotypes has to be occurring in utero, in the placenta. So what is it that's going on? So we looked here, and we found that there was a placental T cell homeostasis which was disturbed in women with HIV. So what does that mean? When we crudely looked and this is completely agnostic of antigen at this stage. When we looked at CD45RA versus CD28, which is a very rough marker of memory maturation in the CD3, CD8 positive population. So this would be naive, this would be early differentiated, this would be late differentiated, and this would be terminal differentiation. And what we found here, if you focus here, is this is in the control group, and this is in the exposed placentas. We found that there was an increased proportion of these late and terminal differentiated CD8 cells in the villus tissue. And I'll explain some of you, I'm sure, will very much know the structure of the placenta, but the villus tissue is the fetal side of the placenta. So the dogma is that you don't find CD8 cells in the villus tissue unless you have a condition of velitis of unknown etiology, which we did not detect here. And it is obviously sub-pathological, but we found these CD8 cells. And if you look in the cord blood, we found that there was a reflection of whatever's occurring in the placenta. And what we posit is that these CD8 cells leave the placenta and, of course, enter the baby through the cord blood. And this is what we're finding here. So the question we wanted to ask, and we wrote a grant about this and got funded so we could do this study, is, well, here are the CD8 cells in the villus tissue. What do they recognize? So we turned to cyclomegalovirus, and for good reason. So CMV is a beta herpes virus. It's a latent virus. And 100% of all our pregnant women are CMV seropositive. Now, they may not be viremic, but they're antibody positive, IgG antibody positive to varying levels, some low and some high, when we look in the plasma. So you can see here that this is South Africa here. I'm not sure what the Mexican data is showing, but I've been told informally that it's very high as well. And India and Bangladesh and Iran all have known hotspots of CMV serostatus of the data that we know of. And here is what I think is very interesting. This is maternal CMV seroprevalence, so it's the frequency of antibodies, versus CMV prevalence. And you can see here in what I've circled up here is where we find South Africa, and there is Mexico. So there has to be some data that almost 100% of maternal pregnant women would be CMV seropositive. So what I showed you earlier with those TCR clonotypes in our control group, actually, it was quite right. Those clonotypes were recognizing CMV antigens interestingly, in the naive population, and I'll come on to that of unpicking what that naive population could be. So the key question we have is, what effect do maternal CMV and HIV co-infections have on the developing neonate in utero? And does that account for this poor health in the HIV-exposed uninfected infant? So you can see here is where HIV and CMV are both highly prevalent, which is our case in South Africa. This balance may profoundly alter what goes on in the placenta and at the maternal-fetal interface. And is this where we look for the answer for this poor health in HIV-exposed uninfected? So this is our ethos, if you like, of what we do in our group, RIECA, at Stellenbosch. And here is the pregnant woman living with HIV and being CMV seropositive. There may not be active virus, but we know that pregnancy is a factor for reactivation of latent CMV. And we're looking in the placenta, and we're looking for poor placental development. And the holy grail, if you like, is can we find a biomarker or a biomarker signature in the pregnant woman that may indicate any of these abnormalities that can give rise to either poor immunontogeny, but as I showed you earlier, poor health and poor neurocognition, for language development. And these are certainly areas that we're thinking about and writing grants, hoping that somewhere we'll get funded to do this. So here, of course, is where we need to look. Here's the discoid human placenta. For those who may not be familiar with the structure of the placenta, let me just briefly take you through it. So here are the decidual membranes, which is the maternal-fetal interface. This is the fetal side of the placenta, and these are the cotyledons, which contain the trophoblasts and the placental macrophages. Very important for vascularization in this region, so that the fetal capillaries can meet the remodeled maternal capillaries that enter through the decidua and never physically meet, but you have, through the intervillous space, this very close contact of getting nutrients and oxygen, as well as immune cells, which are very important in terms of microchimerism and tolerance induction. Because, of course, the fetus is semi-allogenic. So we had the hypothesis. So the hypothesis is that the maternally derived activated CMV-specific CD8 T cells, so these CD8 T cells that we observed in earlier studies, traffic from the blood, maternal blood, to the uterine decidua and acquire tissue-resident and polycytolytic programs, so they express molecules related to their cytotoxic activity, and cluster with the cytotrophoblasts. So what I'm going to show you is everything except the clustering with cytotrophoblasts at this stage. This is unpublished work. I'm being very daring and unusual when most people will only present published work, but I think it's important to show you the ongoing work in progress from our group. So I'm going to jump the gun a bit and saying that these cytotoxic CD8 T cells actually may have a two-edged sword to it. One is control of virus, the other is pathology. So I just want you to keep that at the back of your mind as I progress through. So I have 5 parts to the talk. The first is, well, what are these CD8 T cells recognizing? Which epitopes in CMV are dominant? Then to map the CMV-specific compartment and what HIV does to that compartment. Then I will be comparing in part 3 the difference between maternal blood and the decidua, what we find in the decidua membrane of the maternal-fetal interface. And then part 4, it appears that the maternal epitope-specific T cells reach the decidua, and I'll show you evidence of why we know that that's happening. Not think, we know it's happening. And also what these cells might be recognizing by looking at CMV antigens, and we're using PP65, which is a dominant tegument protein of CMV, as an example of where CMV antigen resides in the placenta. So this is the study that we performed and we got funded to do. So we had 104 pregnant people, some living with HIV and some living without it, but they're all CMV seropositive. And what we also made sure of is that we recruited male babies, the placentas from male babies, because we wanted to know the origin of any of the immune cells that we found in the decidua as well as the villus tissue, which I won't tell you about. And we looked at different populations, and we employed a 27-color panel looking at activation, memory, chemokine receptors, plus using CMV pentamers to HLA A, B, and C epitopes. So for those who are not familiar with pentamer or tetramer technology, tetramer is 4, pentamer is 5. These are 5 MHC molecules, and in the groove are the epitopes that are found in CMV. So these were off the shelf. We bought 8 of them, covering 5 different HLA molecules that are common in the South African population, which were covering probably around about 60 to 65% of the population. So we got many hits as we wanted to predict, and that was fine, and that was great because it allowed us to measure the CMV-specific T cell immunity, and these, of course, are CD8 positive T cells. And because it's biotinylated to a fluorescent core, we can use it in our 27-color panel. So these are the epitopes that are being recognized by those CD8 T cells that we are detecting. So you can see that ranging from HLA-A201 down to the Cs, CO6 and CO7, we find that the epitopes in C that are restricted by OCO2, this particular epitope, which we call the CRV epitope, is highly immunodominant. It means it's being found on, certainly of the 29 we looked at, 17 of the 29 women, regardless of whether they were HIV infected or not. If you take all the Cs, which are the CO6, there's 2 epitopes in CO6, which covers this area here. You can see it's about 80% of the pie. So these HLA-C restricted epitopes are extremely, I think, relevant. And you might, for those amongst us who know this, HLA-C is also contains the KIR ligand, but also, more importantly, the placenta only expresses HLA-C along with HLA-E, along with HLA-G, not HLA-A and B. Many, many publications have looked at CMV, not necessarily in the placenta, but in general, and have restricted themselves to HLA-A0201. Now, had we done that, we wouldn't have come to any kind of conclusions whatsoever. And this is what the pentamer staining looks like. So what is important is that because we could combine it with a 27-color panel, we could identify the memory maturation of these cells. And so these cells tended to be of a TEMRA phenotype, and I'll explain what TEMRA is in a moment. And basically, TEMRA cells phenotypically are expressing CD45RA, which is found on naive cells, but only in the context of co-expression with CCR7 and other molecules, but specifically CCR7. But in TEMRA cells, CCR7 is absent because what's happening is that TEMRA cells are at the end stage of their life, and they are re-expressing CD45RA. And you can see here in this quadrant here that these overlaid dark plots are the pentamer stains, that they appear, this one particularly, which is the CRV epitope, certainly almost 100% of the cells are in this fourth quadrant, which is marking our TEMRA cells. If we collate all of this, we find that the TEMRA cells are very much skewed to the larger response that we identify in these pregnant women. So this is in the antenatal bloods prior to delivery. So the kinetics of these cells is very interesting as well, because it gives us a clue as to what type of memory is forming. Bear in mind that I haven't specifically said this, but we haven't identified any CMV reactivation, albeit looking in the plasma. So there's no plasma viremia. They're very high antibody levels for sure, but no active CMV that we could detect. And what we found, if you look between 31 and 39 weeks antenatally, we find there is a significant expansion of these CD8 pentamer positive cells. And in fact, this HLA-OCO2 CRV epitope restricted response is driving this expansion. The red dot is HIV infection. The darker dots are not HIV, so it's in both. You have this expansion. Whereas the other QIK epitope is not driving this expansion. It's there, but it's not being expanded. And this is very reminiscent of an inflationary memory T cell response that you find in solid organ transplant. It's been looked at in terms of kidney transplantation and CMV reactivation. Plus, in the mouse studies, looking at murine CMV, where there's no virus, but the memory cells are inflated. They just continue going. And so this would be conventional in the green down here, where central memory cells would form if you have a controller virus. The virus is low, either because of drugs like acyclovir for suppression of CMV, or there's natural control. There's a concomitant reduction of these antigen-specific cells. But inflationary is quite the opposite. It just expands. So what inflationary means is that these TEMRA cells that we're picking up, which are at the end of their life, just keep expanding and probably reach an equilibrium according to that part. We don't quite know. But certainly they are expanded by the time, almost by the time of delivery. And these cells are at the terminal end stage where they have recycled CD40, and they importantly are highly cytotoxic. And what I will show you in a moment is that there are different populations of TEMRA cells, but these are the ones to look out for in terms of potential placental damage or placenta control of the virus. So the conclusion to this part is that there's one HLA-C restricted epitope dominates, CRV. These CMV-specific cells sit overwhelmingly in the TEMRA population, which are almost all CRV, epitope-specific responses, and these kinetically expand over gestation from 31 to 39 weeks. So it's really at the very tail end of trimester 3. And there's an expansion without a naive pool, which therefore we think is inflationary. So what I'm going to show you now is we're going to map those CD8 positive, those CRV specific CD8 positive cells, because there are different populations. So what I'm showing you here, if you just look at the UMAP here, there are 15 different cell clusters of CMV-specific CD8 cells, most of which are CRV epitope-specific. And the map spans stem-like memory. So these are the stem cells here, stem-like. I don't think to be confused with mesenchymal stem cells. We call them stem because they're very, very early in their memory maturation. They're cytotoxic, effector, and their terminal CMV. So there are different populations of TEMRA cells. So this provides us with a little bit of a background of what's happening in the blood regarding CMV specificity. What I'm showing you here is the overlay of CD45RA, CCR7, and granzyme B and perforin, which you need both to be cytotoxic. Now, this is likely to be the naive or the very early differentiated stem-like cells because they're RA and CCR7 positive. But this population going down here are CCR7 negative, except for the end here, and some of which are perforin and granzyme positive. So there are almost 4 different populations of TEMRA cells that we can pick up. So not all TEMRAs are one thing, like not all humans, I guess, are one. And what we're doing here is showing you the differential distribution now when we add HIV to the background of this. What's HIV doing? So HIV is expanding the populations of some of these TEMRA cells, the cytotoxic TEMRAs, the stem-like CMV. Remember, these are all CMV specific. And what it's also doing is it's shrinking the terminal CMV TEMRA population. So there's a shrinkage of one population and the expansion of another. So HIV is having a profound effect, almost like black and white, as it were, very, very opposite. So I'm going to talk next about pseudotime, but first I want to explain what that is. Because in our 27-color panel we have memory markers which allow us to look at stem-like memory, which are the very, very early differentiated cells, very early on in CMV differentiation. And then you have towards the end these terminal differentiated cytotoxic cells, and you have a whole shade of memory in between. What pseudotime does is it puts this lineage on a linear path. It's not, of course, time, hence the word pseudotime, but it creates an impression of maturation timeline. And so that's what pseudotime is. So that's how to read it. So it's as I've put here, it's a maturation stage. It's not necessarily the time elapsed. Some maturation stage may go quicker, and some may go smaller, and some maturation stage may stay still. So this is what we're trying to get at here. So what we're showing you here is the effect of HIV on this pseudotime, whether you have a CMV only or an HIV and CMV together. So CMV only is here. This is gray, and it kind of, along with the pink, creates a darker red, and the CMV HIV is in red. So, but I think what I've boxed here is actually what's important, I think, as a take-home message, is that these are CD16 positive, CMV positive, CD8 positive cells. These are NK T cells, the NK CD8 T cells. And CD16 is measuring the FC3 gamma receptor. So these cells could potentially impart mechanistically, linking with the antibodies to CMV and form an antibody-dependent killing, or an ADCC-like reaction, which is, again, a cytotoxic reaction that could be protective, but it also, at the other side, could be pathological. So this is showing you here in pseudotime that it's completely stationary. And this is in the CMV only. As soon as you have an HIV background, you have expansion of these cells. So it's almost like a redistribution of these cells in the presence of HIV. So the bottom line here is that CD16 CMV TEMRA jumps from 0 to 15, way out here, an end state seen only with co-infection. So HIV is having a profound effect on that particular population. So part 2 conclusions is that, of course, there is no naive pool, and they're predominantly TEMRA. And we think what is happening is that there's a very early stage of memory differentiation, which we are calling stem-like memory. It's almost like feeding, and all the cells are stemming from that population. And with only HIV, this CMV TEMRA falls, and every other TEMRA state rises or expands. So what I showed you earlier was all the TEMRA cells, but now we're teasing out what's shrinking, what's expanding. And there is a very cytotoxic NK T cell that's emerging with HIV. So what's now happening at the maternal-fetal interface? And this is where we're getting to the meat of the story that I want to tell you. If you look here on the UMAP here, what's in red is the population of CMV-specific CD8 T cells that we find in the decidua. And you can see that it is completely separate from what we found and what I've been telling you about so far in the maternal blood. And there is a bridge, almost like a connector, and these are the stem cells. And these are the different populations of CMV-specific CD8 T cells in the decidua and in the blood. And these are the pentamer, or the CMV-specific CD8 positive T cells that are found in the decidua, in mostly these clusters here, and in the blood, which is mostly these clusters here, and those of which I have been telling you about. So what are these cells here and that are clustering here? So this is, rather busy, but ignore this side for now and home in on this side. So this is the difference between blood and the decidua one of the decidual membranes. There's two, and this is the peritolus, which is closest to the maternal, the maternal blood. And you can see here very clearly, as the UMAP was showing you, this is now quantifying it, there is a very different repertoire of CMV-specific CD8 positive T cells in the decidua. And I won't go through these, but what I want to show you on this side is the shared populations. And the shared populations are these CMV stem-like cells, which is why we're hypothesizing that these stem-like cells are feeding both decidua and the blood. And again, this is just quantifying the differences between the blood. So these are these CMV stem-like. They're very much the same between the blood and the placenta. These cytotoxic, one of the cytotoxic temera populations, very evident in the blood, absent in the decidua. Vice versa, the activated tissue-resident memory. What I will say at this stage is that none of this is via manual gating. Of course, this is unsupervised clustering of cells. What I showed you earlier on with the plots of temera cells, that was all manual gating. So the two are very complementary, which is very satisfying for us to know that unsupervised computational analysis agreed with our manual gating analysis. And this would make sense because you find activated tissue-resident memory cells in the decidua. Of course, they would be completely absent from the blood. Likewise, these activated resident CMV-specific cells, and these are again, the cytotoxic effector memory, which would not be tissue-resident, and hence they're very common in the circulation. If we apply pseudo-time, like we did before, what it's showing you here is that this population, the stem-like memory cell, doesn't differentiate. Very much like we found that I showed you earlier from the blood. But in the decidua, it's very much stationary. When you get to the activated tissue-resident memory cells, you can see that these cells, and here along in pink and red, or different shades of, I don't know what these colors are, they're more progressive in their memory maturation. So what we think are that these highly cytotoxic temera cells that we found in the maternal circulation are probably lodging in the decidua and becoming either guardians or culprits to pathology. And these are the stem cells which I think are feeding both sides. So in conclusion, this part, in the blood and decidua form almost entirely separate CD8 clusters or territories. These cytotoxic effector temera dominate the blood, and the resident CMV specific are tissue-resident memory cells. And these CMV stem-like memory cells are shared, and we think are feeding both compartments. And the pseudo-time places the decidual, what's happening in the decidua, the resident cells downstream. In other words, they're more early developed than you find in the blood. So, do these maternal CD8 positive T cells reach the decidua? Well, one line of evidence, which is kind of not hitting it squarely on the head, is that the CRV epitope is found at 31 weeks in the blood and in the decidua when we look in the decidua. These are these tissue-resident memory CD8 cells. So these, and this is happening at 39 weeks. So yes, these CRV epitope-specific cells can be found in the decidua, but that doesn't mean to say that the infant is not making CRV-specific. So what we next did is, it's a bit blurry, but we did some in situ hybridization. We used a prime flow assay, and it's looking at the exist gene, which is responsible for promoting the XX phenotype. And remember, I told you we only collected male placentas from male offspring, so that we could do this assay. And then the KDM5 gene ensures that there's expression of the Y gene. So what I'm showing you here in a blurry pink, I'm not sure why it's blurry, is that these are exist positive cells. And these are 3 representative examples in the deciduous membranes. And you can see that there's some low exist expression. And in fact, when we looked at this in more detail, there's exist dropout in our PCR reaction. So when it comes to the flow, I think this may account for this negative population. But by and large, there's nothing here. This is black, both are black here. This is the control female, this is the control male. We wanted to be sure when you have a negative result, like a negative for KDM5D, that you can pick up a positive response. So we spiked some of the tubes, the control tubes, with male and female cells, just blood cells. And this is what, so we knew the assay was working. So this confirms the fact that these CRV epitope specific CD8 T cells are lodging in the decidua from that inflationary response that I showed you earlier in the maternal blood. So this is what we think is happening. You have the cytotoxic CD8 T cells, which are high just before parturition or delivery. And these cytotoxic CD8 cells then migrate into the decidua, which is all I've shown you at this stage. And then we think that they migrate into the villus because that's what we found very early on, 5 or 6 years ago, which we published, which I showed you very early on. So what are these cells doing? First of all, let's conclude. So these CRB-specific CD8 T cells are directly detectable in the decidua. They are KDM5D negative and exist positive, therefore confirming their maternal origin. These maternal CD8 CTLs migrate, that's our conclusion, and in the decidua, they are increasingly polycytolytic because they're a form of TEMRA, they're a form of exhausted at the end stage that is lodging in the tissue. So the last part, which is the last few minutes, I'm going to talk to you about, well, what are these cells recognizing? So we're using PP65 to understand what they're recognizing. So PP65 is a tegument protein, and it's the most dominant protein from CMV. And it ensures that CMV can replicate in infected cells by silencing transcription factors responsible for getting rid of CMV infection. Our CRV epitope is actually found in the IE1 region. So I'm saying to you right now up front that we can't yet connect the CRV epitope-specific responses with their recognition of what they might be recognizing, but we're looking at PP65 simply because it's the reagent we had at the time. So when we look at the CD45 positive cells, which is part of our 27-color panel, which is obviously going to be found on all leukocytes in the placenta, we found that this is the frequency of PP65 in the CD45 positive population. And we found that PP65 is found inside and on the surface of Treg cells, CD8 positive T cells, and NK cells to varying degrees. When we look at the CD45 negative population, which are all the non-immune cells, which would be the stromal cells, the cytotrophoblasts, the trophoblasts themselves, could even be mesenchymal stem cells. I'm not too sure. I don't know, they may roll up when they're isolated. Because I'll show you in a moment that they're quite small cells. But here, these cells are perforin and granzyme co-expressing. They're expressing class 2 HLA-DR, but not CD38 co-expressing. And they're also expressing PD-1 with a little bit of CD39. Some of this gives us a clue as to what these cells might be. When we use image flow and we wanted to identify, are these PP65 positive cells on the surface, S, or inside the cell, P for permeabilized? And they're both. If you look at the merging of the two cells here, or maybe the overlays, you can see it better. You can see that they're both on the surface, PP65, very nice ring, and intracytoplasmically. That's the CD45 negative population in the parotopes, in the decidua. If we then look in the villus tissue, we found these are much smaller cells, again on the surface and inside the cell, and the same picture. So these PP65 antigens are found everywhere. It's ubiquitous in some ways. When we applied pseudotime to understand what lineage of T cells, in this sense, are being infected with or expressing PP65, we find that the very late stage of terminal differentiation, or if you like, these TEMRA cells, are also expressing PP65. If we look at the villus tissue here, we call this the villus dip. There is a significant dip in the red, which is the CMV and HIV co-infection, and we don't know. Is the dip relative to clearance of cells because they're being recognized due to a more killing power of these cells, or is it just that there's less PP65 at that point in time because they're transitioning to the late stage differentiation? When we associate this polycytolytic CD8 T cells with the presence of PP65 on these CD45 negative, there's a tantalizing negative association. But that could mean one of two things. It could either mean that these polycytolytic CD8 T cells are getting rid of PP65 bearing cells, or the PP65 bearing cells are causing senescence and lower frequency of CD8 positive T cells. This is work in progress, as I pointed out before, and something that we need to probe further. So, in summary, and I'm almost finished, PP65 sits inside maternal Treg CD8 T cells. I find it very difficult to believe that these cells are CMV infected, but we haven't measured it yet, so I hypothesize that they are not. But they're also found in CD45 negative, which we think are trophoblast or stromal cells. And these cells we know are responsible for remodeling the vasculature in the placenta. And antigen load peaks late, yet the villi separate mid-trajectory, not sure why. And there is more cytotoxic capacity with less residual antigen, that negative association. So let's try and put all of this together from this biopic that I've shown you. And that is that you have maternal CMV antigen, and what we think is happening is that the antigen is spreading from the maternal circulation to the placenta. And we think that all antigens do this, and I'm not convinced yet that the placenta is a CMV reservoir. It might be an antigenic reservoir. And that may be true for HIV antigens as well, but we haven't looked at that. PP65 antigen is displayed on these CD45 negative cells. These maternal CD8 T cells migrate from these inflationary T cells, migrate from the peripheral circulation into the decidua, and I think migrate even further into the villus tissue, causing one of two things. Either the guardian path and protection against vertical transmission or protection against the placenta infection of CMV, or the culprit path, meaning that these cells are causing damage in the placenta, leading to pathology and impaired spiral artery remodeling. So we generated data. What now is our hypothesis? So it's here. We hypothesize that HIV co-infection drives a Temer-like cytotoxic shift alongside incomplete antigen clearance. And this tips the balance from this path to this path. And almost, last minute, promise, we identified a long time ago that with HIV infection, you have this condition, you have maternal vascular malperfusion. And what we think is, could maternal immunity to CMV be driving this pathology? Where is it? Here it is. So this is a very good example of villus histology. We know with HIV infection, there's a twofold increase in maternal vascular malperfusion. This is a normal villus where these spaces are intervillus spaces where the maternal blood flows. And within the trophoblasts, or these globular-looking cells, are the fetal capillaries. In a condition of maternal vascular malperfusion, you can see that the spaces are greatly reduced and there's enlarged, engorged trophoblasts with a highly red appearance of these fetal red blood cells within the fetal capillaries. And the reason they're looking very bright and red is because they're not moving, they're stationary. So there's some level of stasis. And that is what happens with maternal vascular malperfusion. So, in conclusion, overall, there's one convergent pathway. CMV-specific CD8s mature in the blood and then reside in the tissue. There's one dominant epitope, the CRV. The target and tissue overlap. So it's either culprit or guardians. And HIV shifts the balance towards this persistent activation and altered antigenic handling. And this is a paradox because, and it's the hypothesis, either you have good or bad, and they probably are happening all at the same time. So could that explain the adverse birth outcomes in these HIV-exposed, uninfected children that I told you at the beginning? And I want to end off with this quote. It was made 350-odd years ago, but I think it's relevant for us to understand everything in life. The history of man, bearing in mind it was written all that time ago, the history of man for the 9 months preceding his birth would probably be far more interesting and contains events of greater moment than all the 70 years that follow it, i.e., everything begins in utero. These are my acknowledgements from my group at Stellenbosch University, where I used to be at University of Cape Town. Where is it? Here. Our colleagues at the Mayo Clinic, University of Miami, and University of Toronto. Thank you very much. Happy to take questions if there's time. Yes.
Voy a hacer la pregunta en inglés para facilitar la traducción. Thank you very much. It was very provocative. A couple of questions regarding the design of the study. You had 109 basically entered in the study, and then a subset of those actually qualified or were studied.
Yes.
How many of them were prima gravida?
I cannot remember off.
It's an important question.
It is very important because you've got antigen experience.
How many of them were toxemic?
That I don't know, but yes.
And how many had premature deliveries?
None. They were all term.
And nutritional status?
Don't know. Yeah, good question.
Thank you.
Yeah, but I can't answer the prima gravi question, but we know it's important because many of our women are not prima gravi. they're on their second or more, often their fifth pregnancy.
The age is important because we don't know if either the CMV or the HIV had been ongoing for years or months or
Yes.
recent.
Yes.
¿Alguien más tiene una pregunta?
Thank you for your talk. Do you expect to have similar behavior with another viruses infections, like for example COVID? Have you ever evaluated this type of infections? Or what's the particular characteristic of HIV that may have this impact with CMV?
I think HIV is a driver of T cell exhaustion. That is known certainly from the peripheral blood studies that I've been involved in as well. So I think that's going to happen in the placenta. I think this is going to be happening all the time, even in the absence of CMV, but to other viruses or other antigens. Yeah.
¿Alguien más tiene una pregunta?
Thanks. Extraordinary work. I wonder whether TLR ligation is playing a big role here in the selection of early cell progenitors that express TLR 3, 7, 9, 8, because CMV is a TLR 9
Yes.
agonist, while HIV is 3, 7, and 8 agonist. And if you have looked, I don't know,
No, we haven't, but that's a great...
TLR expression in your early progenitors,
No, we've not looked.
especially in the bipotential progenitors, T and K progenitors,
Yeah.
if they express these TLRs, maybe that expansion you see in the early development or differentiation is devoted to this pathway triggering.
Great suggestion. No, I like it.
No?
I can't. We haven't looked.
Okay. And also mesenchymal stromal cells from placenta are expressing TLR cells. So it could be a bystander phenomenon, not only the direct stimulation of early differentiation.
Yeah.
Have you tried FTOCs, fetal thymic organ cultures? With your progenitors, your inflationary TEMRA progenitors can give you some answers in that.
No, agree. Oh, no, we haven't looked at any of that. But I think it's very important.
And the last
distinguish
question
what's
is,
causing what.
how long do these TEMRA progenitors live? I mean, is long life, or is just an emergency hematopoiesis while they are exposed?
It could well be a pregnancy phenomenon. What we need to do is to look in non-pregnancy, during HIV and non-HIV. Birth itself is an inflammatory process. It could be the natural inflammatory process of parturition drives these TEMRA cells. More than the virus, But good
there is a piece of information about emergency lymphopoiesis, giving the rise of interferon killer dendritic cells, and they are a combination of lymphoid cells, innate lymphoid cells, but with cytotoxic properties. So maybe something like that.
Yeah.
They are restricted. The expected repertoire is not so high. So maybe it's an emergency hematopoiesis.
Giving lots of ideas.
Yep.
Yeah.
Thank you. Thank you.
I do have a couple of questions too. Have you see if your TEMRA high CD28 negative cells, CDAs, are senescence, the whole spectrum of senescence? For example, are they secretory of many cytokines? And have you seen it?
We haven't looked.
You don't know if you have the whole
You're asking me
senescence.
great questions, things that we've not looked
Because
at.
CMV causes senescence.
Yes.
But the number of TEMRA high cells
Yeah.
far exceeds the number of CMV positive cells that you are seeing.
Yeah.
So you might have other viruses that also
Yes,
give
yes.
senescence, like EVV
Yeah.
or HB6 that is
Yeah.
also complicates a lot transplant,
Yeah.
like CMV does.
Yeah. Yeah.
So, and one characteristic of these senescent cells is that they fill with markers of NK cells. So they tend to be
really?
like CDA
Okay.
NK hybrids.
That's exactly what we might be seeing.
Exactly. So
Yeah,
it might
yeah.
be just a CDA senescent cell
Yeah.
and not an NKT.
Got it. Very nice. Thank you. Yeah.
There is a question from internet. Thank you for your excellent presentation. Do you believe that there are metabolic effects of antiretroviral treatment? And if this could induce mobilization of cytotoxic cells to the decidua?
So we have evidence, not necessarily for that last part, but we have evidence that the different antiretroviral drugs in vivo actually reduce the functional capacity of macrophages via the transglutaminase enzyme factor 13A1. So these drugs are doing something potentially pathological, and it tends to associate with preterm birth. So whether they influence the cytotoxic T cell, it's something that we could look at. It's a good point. I don't know whether there's any evidence for that in the periphery or not. But of course, the tissues are so different. Yeah. Yeah. So we know these drugs are doing something, for sure, and not necessarily a good thing. So I think the field is so focused on viral suppression, which is what these drugs do wonderfully, and they prevent, they prolong life, they prevent transmission, but we know they have cardiovascular problems. We know they have lipid dystrophy problems. Well, what they do for the immune system, open question still. But we know that they interfere with macrophage signaling in the placenta.
Another one. Do you think that the retroviral treatment taken by the mother might alter the placenta development?
Yes.
How?
So the macrophages, which are placental, are very instrumental in vascular development. So if you have abnormal placental macrophage function, and we've done some in vitro experiments in Hoover cells where we pre-treat macrophages with different combinations of antiretroviral drugs that individuals take, that pregnant women take. We pre-expose them and we then wash off and we put those ARV-treated macrophages into a Hoover assay. There's no branch formation, there's no vascular angiogenesis. So these drugs are having a profound effect. Whether the pro-drug versions of them in vivo, whether the metabolites of these drugs do the same is the next question. Yeah. So bottom line, yes, they have. And I think that it also explains why even with antiretroviral treatment rollout, these HIV-exposed infants, the women, they still have adverse birth outcomes. It's almost the same as if they weren't taking drug. So why? Must be the drug. Type thing, yeah. Simplistically. Yeah.
Okay, let's thank again Dr. Gray for his excellent presentation.
Thank you. Thanks very much.
Muchas gracias a todos por estar aquí. Y agradecemos